So the last thing we need to discuss is how exactly do the pigment molecules like chlorophyll absorb light energy and render that energy usable to the chloroplast for the production of sugar molecules. In order to get into the details, we have to first talk about how are those pigment molecules organized within the thylakoid membranes of the chloroplast. In those thylakoid membranes, those green membranes, the chloroplast has something that are called photosystems. And photosystems are an aggregation of both proteins and the pigment molecules. The proteins sort of help to organize the pigment molecules. Now, a photosystem has two parts to it. I like to think of a photosystem as a donut, so a full donut, a ring, and then imagine shoving a donut hole back into the inside of a donut. So you've got the donut hole and you've got the full donut. The full donut, the ring on the outside, this part of the photosystem is called the light harvesting complex. So in this cartoon diagram here, you can see in this deep purple color, we've cut through half of the photosystem, so this is half of that donut. The light harvesting complexes are essentially protein in purple and then pigment molecules in green. And the main purpose of the light harvesting complexes is exactly what their name implies. They are the ones that will absorb the light energy from the sun and help to funnel it towards the center of the photosystem, so the donut hole that we've shoved back in there. And the center is called the reaction center complex. And here, again, it's made out of primarily protein. We also have a very special pair of chlorophyll molecules there at the bottom. So let's take a look at how a photosystem functions. First of all, this entire photosystem is embedded in the thylakoid membrane inside of the chloroplast. And when a photon of light comes along and it hits one of these pigment molecules in the light harvesting complexes, if that photon of light is the correct amount of energy, has the correct amount of energy, or is the correct wavelength of light to excite one of the electrons within that chlorophyll molecule to the excited state, then the photon energy will be absorbed by the electron will bounce up to the excited state, freaks out, doesn't want to be there, drops back down to the ground state, and re-emits the energy it had just absorbed from the photon. That energy that got re-emitted gets immediately picked up by an electron of an adjacent chlorophyll molecule. Same thing happens there. The electron of the second chlorophyll molecule will bounce up to that excited state, freaks out, doesn't want to be there, drops back down to the ground state, and again, re-emits the same energy it just absorbed. That energy gets picked up by a third chlorophyll molecule and its electron, and the same process happens over and over again. And this way, the energy, as you follow the black arrows here, will bounce from chlorophyll molecule to pigment molecule to pigment molecule, and be funneled into the reaction center complex in the middle of the photosystem. Here, the energy gets picked up by a special pair of chlorophyll A molecules at the bottom. Again, an electron from this special pair of chlorophyll A molecules gets excited by this energy, and instead of just going up to its excited state, it actually completely leaves the chlorophyll A molecules. This is our first redox reaction where this pair of chlorophyll A molecules becomes oxidized, it loses the electron, and the electron will bounce up here to part of the reaction center complex that's called the primary electron acceptor. So the primary electron acceptor gets this electron that has now this boost in energy. So that's essentially how a photosystem will work. So the next thing we need to do is we need to take a look at the different kinds of photosystems that chloroplasts can potentially have. There are two. They're labeled as Photosystem II and Photosystem I, PS2, PS1. The interesting thing is when we look at the process of photosynthesis, you'll notice that Photosystem II works first and then is followed by Photosystem I. So it's kind of counterintuitive, but they were named based on the order in which they were found. So Photosystem, we discovered that one first, and Photosystem II, we discovered it second, even though they work in a backwards fashion. So what is the difference between these two photosystems? Really, they're identical except for that special pair of chlorophyll A molecules in the reaction center complex. For photosystem II, that pair of chlorophyll A molecules is best at absorbing 680 nanometer wavelength of light. So sometimes photosystem II is called P680 to stress that difference in light absorbance of those chlorophyll A molecules. Photosystem I, for that one, that pair of chlorophyll A molecules in the reaction center complex, they are best at absorbing 700 nanometer wavelength of light. All right, so a little bit different. We call that guy P700. Photosystem I, P700. All right, so now how do chloroplasts use these two photosystems to help absorb light energy and use that energy to make sugar molecules? So the next thing we need to do is we need to understand that photosynthesis, just like cellular respiration, is a flow of electrons. If we step back and talk about cellular respiration just for a second, right? So in cellular respiration, you start out with glucose molecules that have high energy electrons, and the electrons get taken away from the glucose. The electrons are forced to give up their energy to make ATP, and then they end up being grabbed by oxygen to make water. Right? So they start in glucose in cellular respiration, they end up in water with low energy. In photosynthesis, we're going to do the opposite. So we're going to switch. We are going to actually take electrons from water where energy is low. These electrons from water, we're going to give them a boost in energy from sunlight, and then we're going to pack those electrons back into organic molecules like glucose as we try to build them. So what we need to do is we need to follow how the electrons flow through photosynthesis. How do they start out in water? How do they get the boost in energy? And how are they built back into organic molecules? Right, so there are two different ways in which electrons can flow in photosynthesis. They can flow in a linear fashion. They can also flow in a cyclical fashion, depending on what kind of organism you're looking at and under what conditions. So the first thing we're going to do is we're going to look at linear electron flow, taking electrons from water and putting them back into glucose molecules. The linear flow of electrons during photosynthesis is the primary pathway. It involves both photosystem I and photosystem II, and it produces both products of the light reaction. So it produces both ATP and NADPH, right, the full electron carrier. So let's take a look at linear electron flow. These next 3 slides are a whole bunch of text. This is for you guys to use as you're studying. I'm going to go through all that text, but I'm going to use these figures instead to explain how the electrons are going to flow in this linear fashion. All right, so this particular slide, you can look at it as a graph. So on the x-axis here we have progress of reaction or time, and then on the y-axis, think of this as looking at the amount of energy. So if you're low at the bottom of the slide, low energy, and if you're closer to the top of the slide, then you have higher energy. All right, so we start out with photosystem II. Remember, this guy functions first in linear flow. And you've got a photon of light here that hits one of the pigment molecules in the light harvesting complexes. If it's the right photon of light, that'll take one of the electrons within that pigment molecule to the excited state. Electron freaks out, doesn't want to be there, drops back down to the ground state, re-emits that energy. That energy then gets picked up by the next chlorophyll molecule or pigment molecule. Again, an electron from that second pigment molecule will be excited to the excited state by absorbing the energy, freaks out, doesn't want to be there, drops back down to the ground state, re-emits the energy, which then bounces to the third pigment molecule. Again, an electron gets excited, goes back down to the ground state, re-emits the energy, and in this way, the energy is bounced from one pigment molecule to the next until it's funneled into the reaction center complex in the middle, where you've got that special pair of chlorophyll A molecules. Remember, in photosystem II, these two chlorophyll A molecules, we call them P680, right? Best at absorbing 680 nanometer wavelength of light. When this special pair of chlorophyll A molecules absorbs that energy, one of the electrons takes that energy with it and it leaves entirely. Our first redox reaction, the P680 becomes oxidized and the primary electron acceptor up here becomes reduced by accepting that electron. All right, so that's so far where we're at in terms of Photosystem II. Now you can imagine that if this process continues and more photons excite more of those electrons from P680 to go up to the primary electron acceptor, there's only a certain number of electrons that P680 has. And so eventually you will deplete them all. And so we have to find a way to replenish the electrons lost. Here's where the water comes in, right? We're starting down here. We are going to take electrons away from the water, right? Water's got the hydrogen atoms, so water gets split. The hydrogens get separated from the oxygen. The oxygen is not needed, and so it'll leave the chloroplast, it'll leave the cell, it'll be sent off into the atmosphere. The hydrogens, however, are a great source of electrons. There are 2 of them here in these 2 hydrogen atoms, and so one at a time the electrons will be funneled into P680 to replenish the electrons that were lost, that went ahead of them. Okay, so let's think about it maybe from the very beginning for Photosystem II, just one more time. Right, so an electron gets taken away from water and is given to P680, and it sits there and it waits. It waits for a photon of light to hit the light harvesting complex's pigment, bounced around from pigment to pigment until that energy flows into the reaction center complex, hits the P680, and the electron will then absorb that energy and shoot up here to the primary electron acceptor. Great. Where does the electron go from there? Well, from there, the electron is handed off into an electron transport chain. Exactly the same thing as what you have in cellular respiration. The electron bounces down the electron transport chain. You'll notice it's getting lower on the slide, so it's losing some of the energy that it had just absorbed from the light. And as it bounces down, the electron transport chain gives up some of its energy. That energy is used to make ATP. So we've got our first product of the light reactions there. ATP produced via the electron transport chain in chemiosmosis. Again, process is literally identical to what you have in cell respiration. Okay, while all of that is happening for Photosystem II, Photosystem I is doing the exact same thing as Photosystem II. So again, the light harvesting complexes are absorbing photons of light, funneling that energy between pigment molecules into the reaction center complex, where P700 accepts that energy. One of its electrons gets excited up there to the primary electron acceptor. We've got to replenish that electron that was lost from P700 now. Instead of splitting more water, instead, you'll notice that the bottom of the electron transport chain here that is between Photosystem II and I ends at P700. So, the electron that was originally in water went to P680, got a boost in energy up here, fell down the electron transport chain to make some ATP, lands in the lap of P700 to replace the electron that was lost before it. It sits there now and it waits for more light energy to be funneled into the Photosystem I's reaction center complex. Once it gets that energy, it gets bumped up here again to the primary electron acceptor. From there, it gets handed off to a shorter electron transport chain. And this electron transport chain literally just puts the electrons into an NADPH bucket. Right? So NADP plus, the electron carrier that's used in photosynthesis, will pick up the electrons, and it will become NADPH. So at this point, we followed an electron, started out in water, got a boost in energy, and now is being held on to by our photosynthetic electron carrier NADPH. Maybe very quickly, let's go through it one more time. An electron starts out in water, gets removed as the water is split, and goes to replenish electrons lost in P680. The electron sits and waits there until enough light energy from the light harvesting complexes gets funneled in there, it can absorb that energy, and it shoots up here to the primary electron acceptor. That's one redox reaction. That's followed by multiple redox reactions as the electron exchanges hands, falling down the electron transport chain. You'll notice it gets lower on the slide. That means it's giving up its energy as it falls down the electron transport chain. That energy is used to make ATP. The electron then lands in the lap of P700 and sits there and waits again. It waits for more light energy to be absorbed by Photosystem I and funneled into P700. The electron will then absorb that energy, be boosted up here to the primary electron acceptor. From here, it gets handed down a short electron transport chain where it's given to NADP+ to make NADPH.